So yeah, no, I was hired as a environmental, basically worked in the environmental agency to make sure that what they were drilling and all that stuff didn't affect the environment nearly as much. And so for a while there, I was sent over to Russia for about a year or two. Now the issue is I didn't speak Russian, so they gave me this information. I had this whole information written up about how we could make sure that the environment that we're affecting, how we can make sure that it stays clean. But it was all in English. But the issue was that the people I was working with spoke Russian, and so the problem was that I didn't speak Russian. I didn't know how to, and so I called my company and I said, "Hey, I need some help here. You're going to need to send me a translator." They said, "Well, can't really send you one, but there's definitely a translation shop where you can go get translators, and it's like part of the Russian embassy or whatever." I said, "Okay, cool." So I went up there, but the issue was that they didn't have any Russian English-speaking people. So that was an issue. But they did have some Spanish Russian people. I was like, all right, cool. Well, I could deal with this. Because in high school, I learned Spanish. In high school and college, I learned Spanish. So I could speak English and Spanish, right? And so I could talk to the translator in Spanish, and then they could translate from Spanish to Russian. Totally worked out, and so I was able to do it. Totally false story, by the way. No, totally false story. But that's the idea with protein synthesis. That's the idea with protein synthesis, okay? So DNA speaks a specific language. I totally had y'all, too. Y'all were like, all right. No, these aren't notes.
So you worked for no oil company?
I did not work for an oil company. DNA speaks a language in G's, C's, A's, and T's, right? Right, G's match with C's, A's match with T's, right? Straights with straights, curves with curves, or GCAT. Okay, so that's the language they speak. The problem is RNA doesn't speak that language. RNA is a lot like DNA, where it is helical. It's in a circular shape. But the problem is, y'all notice the difference here? Would y'all see a difference?
Single copy.
Yeah. So instead of a double helix, it now has a single helix. Okay. It also speaks a different language. It speaks G, C, A, and U. So y'all know, if you're in French or in Spanish right now, y'all noticing some of the words, they're not super hard, like some of the easier words, just because either you've been exposed to them because you live in Texas.
Means cafe.
There you go, exactly. So we call those cognates. And cognates are words that sound the same both in whatever language you're in as well as the other language. It's because we're all based on the same language. We're all based on Romance language.
Language barrier.
Yeah. So, but the issue is that English and Russian don't sound anything similar, right? Or English and Japanese don't sound anything similar because they're not based on the same language. DNA and RNA are based on the same language, okay? Now, proteins, they don't really read one letter at a time. They read 3 letters at a time. Proteins are a little more advanced.
But wait, I have a question.
And they make up, just one second. And they have a series of what's called amino acids. I just always look at it as AAs, amino acids. So there's a language of DNA, there's a language of RNA, and there's a language of proteins. And DNA can speak RNA language. It can translate that pretty easily. And RNA can speak both DNA and protein language. And so it has to transcribe from DNA to RNA, and then translate from that RNA to a longer, fuller language of protein.
So you said RNA.
Have you ever heard of George Ezra?
Name sounds familiar, but I don't know. So anyways, and you'll notice that we're getting into a very, very complicated structure here. Okay, there's helixes, there's spirals, there's folds, which we call beta sheets. There's all clumps of chemicals, and so this whole section is what we call the central dogma of life. So here's the idea. I'm going to give you some cards, and I want you to work in pairs or singles or whatever you want to work as. And what I want you to do is, there are 10 cards here, okay? Protein synthesis.
Protein synthesis.
Protein synthesis, which, what does it mean synthesis mean?
Is it like synthesis
To do
It's like photosynthesis.
Right. So photo means to use light to what?
Not reproduce. Make sugar.
To make. Yeah. So I lost the band on that one. So photosynthesis is using light to make sugar. So protein synthesis is creating new proteins. Okay. So there's actually two steps in protein synthesis. There's transcription and there's translation. So back to this, transcription is from DNA to RNA, and then RNA to proteins is translation. Did y'all catch that? Transcription is from DNA to RNA, and then translation is from RNA to proteins. So there's 10 cards. What I want you to do is take those cards, and you're going to place those cards in order. Now, there's going to be 5 cards that are transcription cards, and there's going to be 5 cards that are translation cards. What I want you to at least do is go through and try to at least figure out the order of transcription, going from DNA to RNA. That's all you got to do right now. Okay? So take those cards, sort them out, and there's going to be 5 cards going from DNA. Where's the DNA located? In the nucleus. And that RNA is eventually going to transfer outside the nucleus. So you're going to start with the DNA and then figure out how does that DNA become RNA. Okay? So again, we're in the nucleus. We're going to find the DNA. On the DNA, we're going to match up with the complementary DNA to a new nucleotide. The A's are now going to match with U's instead of T's. Okay, again, this matches the nucleus, and it looks something like this. So y'all notice the nucleotides themselves. So this is DNA. This is RNA polymerase. Now notice, how many sides of the DNA is it reading?
2.
Nope. 1. Y'all see how only one section of the RNA or DNA is being passed through the RNA polymerase?
Yeah, but then what's this?
Okay. This is merely the RNA polymerase taking it and separating it out. Okay, so let's go back. A couple of things. You remember how on DNA replication there was basically 2 enzymes? There was helicase, and the helicase unwound the helix, right? Okay, and then the DNA polymerase. Remember, it was poly means what?
2.
More than 1. So DNA polymerase is an enzyme. ACE means enzyme that is going to make more polymer DNA. So again, you take that word. You used to say DNA polymerase. So you take ACE, right? It's an enzyme. It's going to make more what? RNA. If it said DNA polymerase, it's going to be an enzyme that makes more DNA. Okay? So again, take those words and break them down. If you know the prefixes and suffixes, it becomes way easier. Because y'all know the word poly, right? You've seen that in math. You've seen that in probably English. Right? I don't know, maybe not. But take these words and break them down to their parts. And I've always told you that ACE means enzyme. So it's an enzyme that makes more RNA. Notice the RNA only works on one side of the DNA. And essentially, guys, you probably don't know this, but when you braid hair, since I have 3 little girls, I know this thing. When you go through and you braid hair, you have to separate it out into different parts, right, before you braid it. So your fingers are essentially the RNA polymerase, where you're separating out the DNA, you're separating out the hair, and you're going to put those into different pieces. Yeah. Okay. So it's the same idea. So this arch right here, this arch at this point is essentially you separating out the DNA. Okay. So the RNA polymerase comes along, and notice anytime there's a T, there's going to be an A. T, A, T, A. A G, there's going to be a C, and A, there's going to be a U. Now you're saying, wait, you said that RNA now is looking at instead of T's, it's looking at U's. Well, it's true, but anytime there's a T, there's still going to be an A there. If the DNA has a T, there's going to be a U. But if you look at this RNA being made, are there any T's here?
No.
No. It's all U's. Okay? So, what do we call this right here? The mRNA. Each letter does mean something different. So each letter represents a different nucleotide or nitrogen base. A just represents what's called adenine. It's a specific structure. C represents cytosine, which is another specific structure. G for guanine and T for thymine. And then U is for uracil. And each letter represents a different structure, just like how you had like the square peg and the square hole. It's the same thing. You have to put the A's with the T's because those 2 pegs fit together. Okay? I don't know. Do you do any electrical work? You do any because you work? Have you worked on cars or anything like that? Like you ever worked on the circuit board of the car? No, or the fuse box? You done? But on the fuse box, a specific fuse has to fit in a specific spot. So if you have a 20 amp fuse, then that 20 amp fuse has to be replaced with another 20 amp fuse. You can't put a 30 amp or you can't put a 10 amp because the 10 amp will burn up too quickly and the 30 amp won't resist enough and it'll catch your car on fire. So it has to fit in a very specific spot. Does that make sense? Yeah, that'd be a bad idea, right? All right. So the enzyme called RNA polymerase links the RNA nucleotides to form a strand of mRNA. or messenger RNA. Okay? So again, it's going to carry this message. So messenger RNA or mRNA carries this message. Where's it going to carry to? Where is protein being made? Not in the nucleus. We're going to leave. It's in the cytoplasm. What part of the cytoplasm? What organelle? The ribosomes. The ribosome. Okay? Think back to your cell organelles. Remember, nucleus is what's holding the DNA, and the ribosomes is our production factor. Okay? So the RNA, the mRNA, it's a messenger RNA. It's almost like the test you're going to have coming up, whatever day that's going to be, which is next time. So Tuesday. Let's say that I had the key to the test, and I needed to send that key to Miss Morgan. What I would do is I would write that key onto a sticky note. So I have the information, so I'm the DNA. That sticky note is the mRNA. Okay, I'm going to put the key on the sticky note, and they're going to give it to one of my students. They're going to run around the building and go to the other location. All right? Don't worry about it. Okay? But that mRNA is that sticky note. So this piece right here is like the sticky note that's going to tell us how to make the specific protein.
So, I don't know what's the difference between mRNA and RNA?
So mRNA is just a specific type of RNA. There's actually 3 types of RNA. There's this mRNA, and we'll look at the other 2 types here in a second.
So does that door go into another hallway?
Yes.
Does mRNA?
Yes.
Go straight to RNA?
All right. So this mRNA, again, that's messenger RNA, is going to be the template to make new proteins. So, ready? Oh, it's not going to do that. It says once transcription is complete, the new mRNA strand leaves the nucleus and moves into the what? The cytoplasm, where it will participate in the next phase of protein synthesis. So, take a look at your cards. Tell me. The first one we said was D. What's the next card? F. Next card is E as an egg, and then J as in jump, and then A. Okay, here's what I need you to do. That paper I gave to you all, which says transcription and translation, flip onto the back, please. You probably only want to use the top half because you're eventually going to glue this work in. Write the word transcription on the top.
Is everyone supposed to have one?
Can I fold it up so I can use the top half?
Yeah.
I don't have one.
You have it? So again, you're going to write the word transcription, and if you know the spelling, it is right here at the very top. Okay. You're going to do a really quick, rough sketch of the different steps of transcription. Okay? And again, yes, labeling is important. So if it says, like, if it labels the nucleus, make sure you label the nucleus. If it labels DNA, label that. But don't be super, like, specific.
You want us to label where it says cell?
Yeah. Well, whatever. Either way. Okay? So again, do just a really quick, rough sketch of those 5 steps of transcription. So you've got transcription done. So again, if you didn't get these cards done, you can kind of finish up here in a second. But we're going to move on. So we've got our mRNA. And our mRNA has what 4 letters? A's, U's, G's, and C's. Okay? So we've got this mRNA and then mRNA. We're moving on. So the mRNA leaves the nucleus. It's going to go towards this little dot here. What's that dot represent? A ribosome. Okay? So now, translation is a whole other step. This is like what on my little story about going from English to Spanish. We've already gone from English to Spanish. Now the translator is going to take that Spanish and translate it into Russian. Okay? So, but it was my story. I told you I was telling you a story. I didn't say it was a non-fiction story.
Isn't this when it chooses every 3?
Yes. This is when it's going to read every 3. Very good. So yeah, if you notice, as we're getting ready, notice that these are all listed in 3s. Okay? So translation occurs in the ribosome, which is actually in the cytoplasm. During translation, the mRNA strand provides a template for the sequence of amino acids in the protein being produced. Each triplet of the mRNA is called a what? You all see right here? Codon. Okay? So here's how we remember. It's called a codon. You'll see it right there. C-O-D-O-N. Codon. So again, we're going to read the mRNA code 3 letters at a time, and those 3 letters are called, since we're reading the code, it's called a codon. Okay?
It's the code on RNA.
All right? So this matches the complementary triplet called the anticodon on the tRNA. So the codon matches up with the anticodon. Just like how the protagonist goes against the antagonist, is the opposite. No. Which is called the tRNA, which is called the transfer ribonucleic acid. So earlier you said, well, what's mRNA? Well, mRNA is one type of RNA. It's sending the message, and tRNA is bringing the transferring in the amino acids. So it's a different kind of RNA, it's a different structure of RNA, but still is RNA as well. Again, it's
like how DNA is, RNA is kind of like DNA.
yeah. But there's 3 different forms. It's kind of like there's 4 different blood types. It's all blood, but you could either have A blood, type B blood, type AB blood, or type O blood. And so there's 3 different types of mRNA, or 3 different types of RNA. There's mRNA, which carries the message. There's tRNA, which carries the amino acids or transfers the amino acids. And there's rRNA, which actually makes up ribosomes. So this ribosome right here, this little piece, this little blue guy, is actually made of RNA as well, RNA and proteins. Okay, so the anticodons of the tRNA match with specific codons of the mRNA. Creates a chain of amino acids. And amino acids are the building blocks of proteins. So they're the monomers of the polymer protein. Okay, so again, you got to kind of keep this in your mind that the monomer for nucleic acid is nucleotides. The monomer for proteins is amino acids. The monomer for lipids is fatty acids and glycerol. The monomer for carbohydrates is monosaccharide.
What makes it up?
Don't do that, please. Is that your ID? Yeah, don't do that, please. Thank you. It like jacks it up severely. Thank you. All right. So if y'all notice this, okay. So we've got this proteins into this translation. What you're going to do is you're going to find, what do we call this? The 3-letter code right here. It's from the code. So this is the RNA codon, and these are called the anticodon. So if our codon reads AUG, what are we looking for?
So the codon is on this.
It would be TAC, but remember we're looking at RNA.
U.
So it's going to be UAC. So UAC. All right, y'all see how it is? So I've got UAC matched up with AUG. This is our codon, this is our anticodon. So
It takes the place of the T, so
yes.
it matches with the A in RNA.
So we're going to match those 2 together, and notice what happens. It moves the ribosome down, or actually moves the RNA to the left, and it keeps up methionine. Okay, this is the amino acid. So the next one I have is GUA, which is going to be, there you go. So CAU is right there. We're going to bring it down to our ribosome. All right, now look, the tRNA went away, the amino acids stayed there, because what we're making here is a chain of amino acids, which is called a protein. All right, CAU is going to be
GUC.
So what's going to happen with the leftover strand of RNA?
We'll see. Let's make our protein, and I'll show you what to do.
So wait, it got rid of the DNA?
It didn't get rid of the DNA. Remember, the DNA is in the nucleus. Okay, let's flash back to where we're at. Okay, the DNA is in the nucleus. That DNA was used, RNA polymerase used the DNA to make mRNA. Okay, that mRNA is right here. That's what this string is. Okay, that mRNA left the nucleus, went to the ribosome, which is in the cytoplasm. The tRNAs are bringing these amino acids to the ribosome. And when I bring it, when I physically click on it, and I grab it, so AAA is going to be what? Okay, U. So when I physically grab it, what I'm doing is I'm basically being like the tRNA. It's going to float itself over here. It's going to link on to that codon, and it's going to move on.
So the tRNA brings it to
Yes, the tRNA brings the what?
I
What are these called?
Anti-
Amino acids.
amino acids.
So amino acids to build the protein. Sure.
So, is the codon, is it always at the bottom of
UUG. ACA.
All right, now, before I put this last one, y'all see this last one that says UAA? All right. That's going to code for what's called a specific stop codon. Okay? If y'all flip into your book on page, let's say, so y'all flip into your book on page 370. Just leave it out right now. Okay. So page 370. There's this thing called a codon chart. Y'all see that? Y'all see the codon chart on 370?
Yeah.
Yes. Okay. So what that codon chart is, and I'll show you all this again, just to keep this out. Basically what that codon chart is these are all the possible amino acids that are allowed. Okay? There's 20 of them. There's actually a couple more, but they don't fit on this codon chart. They're weird ones. But essentially we have 20 amino acids. Those amino acids are read by these 3-letter codes. So very easily you can go through and you can look at the first, second, and third letters, and those will then tell you exactly what amino acid is going to link on there. All right? Now, what do those yellow ones represent? The starts and the stops. Now notice the start is only methionine. Okay? So every single protein you have in your body starts with methionine. Okay, because that's the start codon. That tells you, okay, at this point, this is where you're going to start. But now, not every part of your body is going to end with the same stop codon. Now, what's going to happen, let's say that one of our codons was supposed to be UAC, to be tyrosine, but it was misread, and instead of it saying, and there's like 5 or 6 more codons down the road, but instead of saying UAC, it's going to read UAA. What's going to happen? It's going to stop. Are those other 5 and 6 going to be read? No. So all of a sudden your protein is now too short. And so let's say that this protein was supposed to make the insulin. Okay? And so now your insulin is not the right shape and not the right size. Is it going to be able to break down into glucose anymore? No. And so then at this point you have diabetes. That's how you get things like genetic diseases. Now, that's not exactly how diabetes works. Diabetes is something else. It's actually, I believe it's a regulation on sending the reception proteins on the outside of your cell, but it's very, very closely related to it. Okay? So these mutations we were looking at earlier, this is where that comes into play. Because remember, DNA makes up RNA, and RNA makes up protein. And if your DNA is messed up, then that means your RNA is messed up, and that makes your proteins are going to be messed up. Does that make sense? Okay? So it all works. So if your DNA is messed up, your RNA is going to be messed up. But if your DNA is fine, but if your RNA gets coded wrong, then your proteins are going to be messed up. Okay? So there's a bunch of different little locations, and it's very specific in the sequence of DNA, and it's very specific in the sequence of amino acids. Everything else, lipids, it doesn't matter if the lipids are in the wrong order. If the glucose is in the wrong order, it doesn't matter. But if your A's, T's, G's, and C's are in the wrong order, or if your A's, U's, G's, and C's are in the wrong order, or your amino acids are in the wrong order, it's a big, huge deal. Question?
What if everything is right with the proteins?
The proteins could actually be folded wrong, which does happen. Then you're still going to have some kind of abnormality, some kind of disease of some sort. Okay, so like sickle cell anemia. Sickle cell anemia is about 121 total amino acids. It's where instead of your blood being in a round shape, it's in a sickle shape, which is a kind of a crescent moon, a banana shape. Okay, and what it is I think it's on amino acid number, I want to say 60, 67, something like that. It should say valine, I want to say, but it says lysine, and that one amino acid change, only that one change, causes it to where the folding of the protein, instead of folding in a nice round globular style, it actually folds in a very flat squish style. And so that flat squish style can't hold the heme, which is what carries the oxygen, the iron. And so if it can't carry oxygen, then you have sickle cell anemia, and that sickle cell anemia causes things like muscle cramps, and it causes things like heart attack and clogged artery and all kinds of other things. So it's just one change that causes a whole other condition. Okay? All right, so let's take our cards. What's our first card going to be? Which one? I? I think that's the last card, actually. We've done D, F, E, J, and A, right?
What is this? This is.
So we're doing translation. H. So the first card's going to be H. The mRNA molecule binds to the ribosome. And then B, as in boy. The tRNA molecules. You'll see how on H, it's just the RNA. Okay, and the RNA has here's the ribosome. And then it says the tRNA comes and binds to those codons. So the next one is going to be G, as in golf. The mRNA continues to move along the ribosome, which we just saw. After G is going to be C, and then I. All right?
Put it backwards from where I had it.
Yeah. So, go ahead and write down translation. Translation is right here. Okay. So write down translation, and then do a quick sketch on those cards. So on the front of that page, transcription translation page, it says DNA replication. So again, we're going to relate all these together. So DNA replication, what happens? Yeah, so what does that mean? What's the layman term? Yeah, copy DNA. DNA. Right? So we're going to copy DNA. That's all. What subunits are used to make the DNA? What are the two enzymes we have to use? Okay, so one of them is DNA polymerase, and I'm writing down the bottom for a reason. So I would suggest doing the same. So this is on the front of that sheet. So DNA polymerase, and then so DNA polymerase makes more DNA. What unwinds the helix?
Double helix. Helicase.
Helicase, sorry. Okay. So helicase. Oops. Not helicase. Helicase. Okay. Now, where does this happen?
In the ribosome?
No. Where's DNA found? Nucleus. So this has to happen in the nucleus. Nucleus.
No, I was looking at
Polymerase. Poly. Yeah, it's
Is that a cursive word right there?
polymerase.
I know you like made it cursive.
All right. And then what's the end product of new DNA? Is it just random new DNA?
No, it's the same.
It's identical. So identical copies of DNA. And I'm going to shorthand this, but this is what it means. So it's identical copies of DNA, and it has two new copies. Now, each copy, there's going to be an old strand bonded to a new strand.
It's gonna have two.
It's going to have an old strand bonded to a new strand, and then the other copy is going to have an old strand bonded to a new strand. And we talked about that when we did the little paper thing we unfolded. The original and the new. Okay?
You also, for every new strand,
Yes. And homework as well. Yep. Okay. So transcription. What's the goal of transcription? What happens? Remember, we're going to do what to what? We're going to start with DNA, because DNA has the code, and it's going to become what?
RNA.
RNA.
And it, you stop right there.
There you go. That's all transcription is. The goal of transcription is to take DNA and make it into RNA. All right. What do we need for that to occur? Only one enzyme. No. It ends in an ACE because it's an enzyme. RNA polymerase. Because we're going to make more RNA, and the way we make more RNA is by an enzyme. And since it's an enzyme that's going to make more, we call it a polymerase. Since it's making more RNA, it's an RNA polymerase. So where does this happen? Where's the DNA located? In the nucleus, so it has to happen in the nucleus. DNA does not ever leave the nucleus.
so but the RNA, what's happening in the ribosome or whatever?
The ribosome is where you make RNA makes proteins, which is translation. Okay? So what's our end product? What do we make? No? What needs to leave the nucleus? RNA. What kind of RNA? mRNA. So mRNA. That message, that sticky note, needs to leave the nucleus as mRNA. Okay? So that's transcription. So transcription is DNA to RNA, then translation's got to be.
What happens if we tell our parents to come get us? Will we get in trouble?
No.
Only if they say something.
They used to try to threaten us to laugh.
Who's in it?
Mutation, which is insertion. You'll see where it says TAC, CTA, right? Yeah. What it's going to do is basically it's going to insert another amino acid in there. So let's say right there they insert a G. So now no longer does it say TAC, GC, TA. What it's going to read now is it's going to read like TAC is going to be AUG, right? Yeah. And it should have gone AUG, GAU, blah, blah, blah. Okay? What's going to read now is going to read AUG. That G has to be read, so it's going to go AUG, CGA. And it's going to shift everything to the right. Y'all see how that would work? No. So basically, if I put someone right there, like if on this front row, there's 6 chairs, right? Or like on the back row, everyone's in there, right? But I grab Brendan and I throw him in the back row. It's going to make everyone shift, right? So no longer are you in the same place. But if I go in that back row and I pull Mackenzie's chair out and I completely destroy that part of the thing, what's going to happen? What's going to happen to the table? It's going to shift together. Okay. So no longer is it going to go is it going to go Brenna, Mackenzie, Jacob? We knock Mackenzie out. We screwed everyone over. Okay, it's a deletion mutation. But if I take Brennan and I throw him in there, that's an addition mutation. Okay, or an insertion. The substitution means that if I take Brennan and I pull Mackenzie out, but I leave her chair there and I put Brennan in her chair. Y'all see that? That's a substitution. Okay. The last one is what's called a three-base insertion. That's what if I take these three in the front and I throw them in the back. Okay? And actually it's more correctly if I take Brennan, Mackenzie, and Jacob and I make a copy of them and I put them in the middle and I make another copy and I put them in the middle.
But this only happens in RNA, right?
It's only, well, it happens in DNA. It happens in the DNA.
It can.
It happens in the DNA that then affects the RNA. That's what I said. Now go ahead and bundle these up.